It's already on.
Good afternoon, everyone. Welcome to the International Seminars on Health, a course offered by the postgraduate program in nutrition at the Federal University of Santa Catarina. I am Ana Carolina, one of the professors of this course, together with my colleague who is here, Deborah Higger, and with our postdoctoral fellow, Mariana Kremer. We are happy to have you join us today for the first day of our seminar series of this year. Our goal is to foster connections between postgraduate students and professionals in their respective fields of interest. And for each seminar, you have a presentation by the guest speaker, lasting between 15 to 30 minutes, followed by a 15-minute question and answers session where you all have the opportunity to ask questions and engage in a discussion if you want to. The students of this course have carefully curated a diverse lineup of esteemed guest speakers who are references in their areas of expertise. And today, our student Danielle will be hosting Dr. Brian Christie, who is here, and Laura Volpato will be hosting Dr. JoAnne Arcand from Canada, both from Canada. And JoAnne Arcand will be with us at 2 p.m. in Brazil. Now, I would like to ask Danielle to introduce our first guest, Dr. Brian Christie. Please, Danielle.
Thank you. Hello, everybody. I'm kind of nervous, but I hope that we have a really great time. So, I'm Danielle. I am a master's student here at the University Federal of Santa Catarina, and it's a pleasure to introduce our guest professor, Dr. Brian Christie. Dr. Christie is a professor of medicine and neuroscience at the University of Victoria in Canada. He completed his bachelor and master degree at the University of Calgary. He's a PhD at the University of Otago with Professor Clave, and he did his postdoctoral training at the Baylor College of Medicine with Professor Daniel Johnston, and he in the Salk Institute for Biological Science. He's an active member of the Society for Neuroscience, Canadian Association of Neuroscience, Research Science and on alcohol, fat alcohol spectrum disorders, research group, and BC Conclusion Advisory Network. He is currently focused on synaptic plasticity and neurogenesis in the developing and aging brain can be harnessed to mitigate the effects of trauma and substance exposure, genetic disorders, and aging. His academic path is both distinguished and inspiring, and today he's going to share his professional journey and current perspectives in his field with us. So, Professor, welcome again, and you have 20 to 30 minutes to your presentation.
Thank you so much for the kind introduction. I appreciate it. And I hope to share with you, some of the journey I've had so far to get to my current position, and to enlighten you that it's not all planned, that there is a lot of serendipity to this journey that took me from where I grew up in the top left. You can see the Indian chiefs. I grew up next to the Sarcee Reserve in Canada. I'll tell you a little bit about that, and went from there to University of Otago in New Zealand, which was a considerable distance away, before finally ending up here in Vancouver Island, another island, much like Florianopolis. It's one of the beautiful places in Canada to live. And so we've had a lot of different things happen in my life to get me here, and I'll try and share that with you. So the first thing I want to define is, what is serendipity? And this is when you often have an unsought or unintended or unexpected but fortunate discovery or learning experience that happens by accident. And Louis Pasteur, said, in the field of observation, chance favors only the prepared mind. And this is, I think, very important for serendipity too. You can have fortunate or lucky things happen to you over your career, but you really only capitalize on it if you're aware and you're thinking about what's going on. And I didn't always think about what was going on, and I'll share that with you too a little bit. So where I grew up was in a part of Canada known as the Treaty 7 territories, and this is in southern Alberta. It's right where the Great Plains meet the mountains. So this view on the right here is actually looking out across the plains, and this is the center, the city of Calgary. And where I grew up is right around here in this little red area, and you can see there's not much there. I used to hunt. I used to run around with a rifle and hunt in this area. These are gravel pits down here that the city used, and I would ride around on my bike, basically not watched. My parents never knew where I was during the day. I lived right next to the Indian Reserve, as I said, so I spent a lot of time on the reserve itself, and that really influenced me as I was growing up. I was quite adventurous and willing to take risks. But I was also sort of a normal child for the time. Calgary is known for the Calgary Stampede, and so at one time of the year, everyone dresses up like cowboys, and we go to a fair where there's lots of food and games and rides. I played lots of sports growing up, and because I came from a very poor background, sport was one of the main things that a lot of me and my friends engaged in. It was the only thing we really had for us, and surprisingly early on we were really good at soccer or football, for Brazil, one of Brazil's favorite games, and we did lots of running, chasing the ball, very poorly conditioned fields, lots of cuts and bruises when we played, but we really enjoyed ourselves. And I think because of this early sporting background, I also ended up developing aspirations to play football. And in high school, I became very good at playing American football, where you wear the pads and the helmets and that. And actually only ended up going to university because I was really good at this game. No one in my family had gone to university before me. I was the first one to go, and I really only went to play football. And before the first season started, I ended up with a knee injury. And even worse, I actually ended up with 2 knee injuries. And so before I even got to play a game, I was in rehabilitation. And my knees were never going to be the same again, and it took a long time to get there. The only sort of interesting thing for me out of this time was that I was one of the first people to get electrostimulation in Canada to help rebuild the musculature around the knees. So they put these little electric pads on my knees and then turned on current. And it wasn't very precise back then. And so sometimes my legs would, all of the muscles would be activated, and I would see muscles that I didn't even know I had because I would have this electrical current flowing through them. And it was painful, but also interesting to go through the process, and maybe that helped drive my interest in what was to come. So, but before I get there, I'll tell you what school was like for me. Because of the knee injury, I had to sort of reinvent myself. I had always seen myself as being an athlete and only went to university to play athletics. I wasn't there to go to school and become educated. And I was, stuck in these large classes with lots of students. I did not do well. I was failing my first year, and my GPA was 1.34 out of 4. That's on the failure side, below 50%. And I was ready to quit university and go become a mechanic or something like that. But my grandma was very happy. She was very happy that I was at university, and she told me this at Christmas. And so I needed to go back. I felt I needed to go and please her. So her little baby had to go back to the university, and I had to learn to navigate the labyrinth of education and find what worked for me. And I wasn't always successful. I did some unusual things. One of the first things I did, because I had problems walking and still wanted to be active in sports, is I joined the rodeo team at the university and ended up riding bulls. I don't think I was thinking too well at this time. But I did that. I spent a lot of time out, made new friends that were engaged in interesting occupations. And so we say in Canada, I kind of grew my hair out and became a different, type of individual for the 1980s. And this was in the 1980s when I was in school. But along that journey, I met a professor, and his name was Dr. Brian Bland, and he taught a course in biological psychology. And one of the main things he gave me was this idea that science could actually be fun. He was always so happy and so positive about anything to do with science. And his laboratory was interested in this EEG waveform called hippocampal theta activity. And he used to come and he would be almost mesmerized by this wave that you could record with EEG. And he was particularly interested how the cells in the brain fired, or their unit activity came together to create these large EEG waves. So the waves are representative of the activity of hundreds, if not thousands of cells. But he was interested in looking at individual cells and seeing how they contributed to the formation of EEG. He also was really interested in the toys in his laboratory. So he loved playing with the electrophysiology equipment. And there was a lot of equipment in his laboratory. And it took a long time to learn what each one of these pieces did. I now can look at it and know that this is a polygraph on the left, an oscilloscope on the right. This is a digitizing unit above here. And this is a video tape recorder. And I'll talk about that in a second. But he really brought this love for the toys of neuroscience to me. And I think that happiness was something that really kept me going, because the course that I was in with him really involved learning how to do small animal surgery. And so I learned how to do stereotaxic placements, recordings, injections, etc. And I didn't find this much of a challenge. In fact, I started working with Dr. Bland because I was very reliable at doing these small animal surgeries. But the challenge was actually taking any recordings and analyzing them. So I mentioned there is this tape recorder in the laboratory, and this VHS tape recorder was how we recorded our data. There were no computers in the laboratory at this time. So we recorded all of these signals on a VHS tape, and then we had to transfer that into a computer that was in one room in the building. And at this time, these PDP-11 computers were the main computers around, and this individual here is actually pulling the disk, the electronic disk that holds all of the data out of the container, and he's going to insert it into this computer. Now, this is the size of a huge platter, and and they were actually called platters, and that's what we were put into the machine. And these big disks held maybe 10 megabytes worth of data. So you could get maybe 10 pictures from your iPhone onto this disk. That's how much data it held. So there was a lot of effort put into this. And I have here on the right that to run one analysis usually took about 15 minutes. This would be run in about 1 second on a modern computer. So there's a huge difference in capacity from then to now. But we managed to use these computers, and it helped us start determining how cells in the brain would fire to make up EEG waveforms. And we started building up this idea of circuit dynamics. And this is, I think, some of the early work in circuits. And in Brian's laboratory, what we found is that a lot of the cells either would turn on when there's theta activity. And you can see here's one that bursts, and he called these phasic theta on cells. So they fired in bursts in relationship to the theta waves. And then there were also tonic cells, and they just increased their firing whenever there was theta activity. and they were contributing to a change of the level of depolarization in the brain. But there are also cells that turned off, and these he called theta off. So there are cells that fired in a tonic way, so they fired continuously until theta came on, then they were completely silent. And you had other ones that fired in bursts, except for when theta occurred. And so he basically started a classification scheme that said there are some cells that are really related to the production of theta activity in the brain, and some that aren't. And this led to my first master's thesis, which was developing an appreciation for how the hypothalamus projects to the septal region of the brain, which then projects to the hippocampus, and how these three regions can contribute to the production of theta waves. And what did I get out of that? Well, I developed a lot of small mammal surgical skills, learned how to work with anesthesia. I learned how to do electroencephalography, how to do single unit recordings, and computer analysis. And of course, I developed this love of scientific equipment that I had, or that Dr. Bland had. So when I came away from that, I always forget, he had this sign on his door that I always used to love, and it was, "He who has the most toys wins." And that really was kind of, one of the things he loved so much about the equipment. The more equipment he had, the happier he was in the day. And that, was a joy he really shared with me. So when I finished my master's degree, being the first person in my family to both finish an undergraduate degree and then a master's degree, I didn't know what to do next. And so some of the options that were going through my mind at that time were, do I become a policeman? I thought maybe I could go join the police force. I might have a better chance at getting on because I already had a degree. I was also interested in becoming a fireman. Those were really good jobs back then. You work 7 days on, 7 days off. They paid well. A lot of my friends' parents were firemen, and it seemed like a great job to have at the time. Or do I travel 13,000 kilometers away to another country to do a PhD? And I only came up with this last option because I was reading a magazine called Nature. It was a scientific magazine. And back then there was no online processing. Everything was hand delivered, and you had to flip through the pages of a magazine and read things. And there was an ad saying PhD scholarships available in New Zealand, apply here. And so I ended up applying and sort of taken this philosophy of ondente fortuna iuvate, and that really means fortune favors the bold. And I thought, why not? If at the worst, if I don't like it, I can always come home, and then I can go become a policeman or a fireman. And so I applied, and I was accepted by this individual. His name's Dr. Neil McNaughton, and he brought me down to Otago, which was beautiful. This is the main university building, and it's over to the right of this picture on the right, but the two arrows point at where my first office was, and the second arrow up here is where we did all the experiments. And you can see these are just old Victorian houses. This is where the university's research facilities were housed at this time. And these have since been taken down and they've put up modern buildings, but this is what it was like when I went there. And very different conditions to what I was used to in Canada, where we were in, large buildings like you have at UFSC. Dr. McNaughton's research was focused mainly on administering compounds or drugs to animals and recording how it changed their behavior, their learning and memory. And I was probably as interested in it as the rats were. The rats didn't really like doing the work, especially when they were injected with compounds that were had any analgesic qualities to them, like ketamine or NMDA antagonists, etc. They would tend to go to sleep in the middle. So it was a very boring project for me to be engaged in, especially after I was used to playing with all the toys in Dr. Bland's laboratory. So I quickly found that I didn't have a passion, and watching rats run around in a maze just wasn't doing it for me. And what was really good for this period of time is that Dr. McNaughton also recognized this and encouraged me to look at where my passion actually was. And in the laboratory next door was another professor who was newer there. His name was Dr. Wycliffe Abraham. And he had a passion for looking at electrophysiology, like the laboratory I'd been in before. And he was telling me that he liked the fact that I already knew how to do in vivo surgeries, and was wondering if I'd be interested in a project he wanted to do where he looked at not one, but two pathways into the hippocampus and stimulated them at different time points. So he could see what happens to both pathways, not just one. Most people were only looking at one pathway in the brain at this time. So this was a bit of a challenge. And so we had to figure out how to both stimulate and record two pathways simultaneously in vivo. And he wanted to look at two different forms of synaptic plasticity, both long-term potentiation and long-term depression. And for those of you who aren't familiar with these forms of synaptic plasticity, I'll just give you a little prep on this. But basically, if you stimulate in the brain in either pathway, you're going to generate a response. Okay, and so this is the response here on the right. And this response is primarily carried by AMPA receptors that are being activated. And if you activate enough, you can also activate NMDA receptors in the lateral half of this curve. And my work later on is going to show that NMDA receptors are actually very important for synaptic plasticity. But what I knew at this time was that if you stimulate this pathway over and over again, you can get these very stable recordings. And so the size of the response doesn't change a lot. But if you apply a brief burst of high frequency stimuli, you can actually increase the size of the pathway that you stimulate. Now I'm stimulating two pathways. And so the other pathway, I didn't do anything to. I didn't deliver any high frequency stimuli to it. And what happens to that pathway is it actually depressed. It showed a reduction in synaptic strength. So this is how we studied two different forms of synaptic plasticity. We would basically potentiate one and depress the other. And what we were able to show is that if you blocked LTP with NMDA antagonist, you also blocked the induction of long-term depression. So it gave my very first paper in Dr. Abraham's laboratory was showing that both LTP and LTD had an NMDA component to them and required NMDA receptors to be active for us to see both. And the basic philosophy of a lot of the work I did with these two pathways was to use this medial perforant path to study LTP and the lateral perforant path to study LTD. And we were able to show that that LTD was NMDA dependent. It required calcium influx into cells. And we also showed that you could induce LTP in vivo and have it last up to a year in animals. So we were able to record animals every month for a year and show that this LTP could last for a long period of time. And these were important studies at the start of the field of synaptic plasticity to show these types of things. And in all, during my PhD, I ended up getting 8 publications in the 3 years that I was there. But it was really one publication in particular that really helped my career progress. And this came from some work by a pair of researchers, Patrick Stanton and Terence Sanowski. And they had showed also with the two pathway model that they could get an associative form of long-term depression in the hippocampus induced by something they called Hebbian covariance. And what they meant by Hebbian covariance is that instead of taking the route I did, which was to stimulate one pathway and not the other one and get LTP and LTD, they stimulated one pathway with high frequency and the other pathway they gave a single pulse only when that pathway wasn't being activated. And their idea was, going back to that theta story with Dr. Bland, is that this would induce the theta wave. And then when the trough of the wave happened, you would stimulate. And this should be when all the cells that were active are hyperpolarized. So now your input onto these cells can activate the postsynaptic cells. And so this is anti-Hebbian or well, he called it anti-Hebbian association initially, to mean that the input isn't activating the cells anymore. And so Hebbian synaptic plasticity means the input to a set of cells will activate it. And if those cells can't be activated and the input arrives, then that input should depress, according to Stanton and Sinowski. So this was really interesting work at the time, and it was really the start of computational neurobiology's contributions to synaptic plasticity. And so I ended up doing their study, but I also introduced a control experiment. And because they were doing these individual pulses, I had to run some animals where I only ran nothing in the medial perforant path and gave these single pulses to the lateral perforant path. Now, I'm a young 22-year-old graduate student, and I spent an hour setting up these experiments, running them to get no change, because this single pulses didn't actually produce any change in these pathways. So I started using the animals for additional experiments where I ran either these non-associative or associative forms of synaptic plasticity. And I was very careful to document what I was doing and to randomize whether I gave associative or non-associative stimuli after giving these single pulses. I basically didn't want the animals to go to waste and spent a lot of time investigating them. And I was very careful and documented it. And what I discovered was that if I looked at these traces, if there was no pulse interleaved, I didn't have any effect. But if I looked at when I applied that associative stimuli, if I primed the synapses with these individual pulses at theta frequency, I would get twice as much LTD as I would normally see in these conditions. And this ended up being called priming of associative LTD. And I think maybe because I was attuned to the idea of theta activity in the brain, what I did is I looked at what frequencies of priming could produce more LTD. And what we found is that 5 hertz or theta activity priming was particularly good at increasing the amount of long-term depression you could see in the lateral perforant pathway. And so we ended up with my first major paper, which was priming of associated LTD in the dentate gyrus by theta frequency synaptic activity. And this appeared in the journal Neuron. And I didn't know much about journals and what was important and what wasn't at the time, but my advisor did. And he told me this would be a great journal for us to publish in, and it was. And it became one of my top papers in my early years. It also ended up attracting attention. And one of the individuals that had read it and was interested in it was this individual, Dan Johnston. And so I ended up doing my first postdoctoral position with him. And he was an interesting fellow. He was actually an electrical engineer who entered into neuroscience. So he really had a lot of interest in the toys that were, prevalent in electrophysiology. And when I arrived at his laboratory, he said, would you be interested in trying something new? And I'm really interested in doing fluorescence imaging and whole cell recordings. And I said, that would be great. What I didn't know was buried in this subtext, and you're also going to have to get married. My wife, who came with me from New Zealand, actually had a little visa fiasco. And so it ended up resulting in us getting married the first week that I was in Dan's laboratory. And they were very kind and very supportive of us. And I have very fond memories of being there. But I also have fond memories because Dan gave me a bunch of equipment. And while you see this all together in one piece, this is a microscope that was on the floor of a room, same with these beams and all these things. And he said, well, here's a rig. I want you to use ex vivo slice to do recordings from, fill individual cells with fluorescence dye, and then record how the calcium transients change. And I'd never done electrophysiology in slices before. I'd only done in vivo work. So I had to figure out how to put this together, how to work and mount a camera, how to add in all the extra equipment that goes with it. We did have computers at this time. So we could record responses on the computers rather than the old tape drives I was using back in Dr. Bland's lab. But all of the equipment also had buttons on it. So we weren't really controlling the equipment with the computer. It was basically used to just record the data. And I would adjust the parameters on the instruments themselves. So these are older versions of the AXO patch and AXO clamp amplifiers that some of you may use. And these are stimulators that allow us to change the size of the pulse that we're administering to the animal or to the brain tissue. And one of the things I was most impressed with myself at this time was I figured out a way to actually heat the objective of the microscope to keep the brain tissue warm at 30 degrees so that we could get more accurate responses. They are more real and representative of what you should see in the brain. And so what we saw were these calcium transients. So here you can actually see this is a cell. It's a reverse image. So normally they would be bright white. But I've got an electrode that's going in here, and it's filled the cell with a dye, and we see the reverse image. And we would put these little boxes over various parts of the dendrites and then record how much calcium came in when we fired a bunch of action potentials in the cell. And you can see that you get big transients in parts of the cell. These are actually closer to the soma. And as you move out away from the soma into the distal dendrites, the transients get much smaller. And then what we did is we put different antagonists for different types of calcium channels. This is conotoxin. So it's a snail toxin that's very toxic to humans into our solution so we can block specific types of calcium channels. And that allowed us to map out what calcium channels were active across dendrites. And so here's a picture of what a cell actually looks like filled with it. And you can see the boxes going across. And as you move out, you can start with these individual blocks, get an idea of what proportion of the entire calcium signal is contributed by different calcium channels. And this was actually a surprising finding because everyone assumed that calcium channels and all receptors in the brain would be uniformly distributed throughout the dendrites of cells, but there is actually a change. And as you went out further in the dendrites, something called low voltage activated calcium channels tended to be more prevalent, and closer high voltage calcium channels tend to be more prevalent. So your cell had to be depolarized more closer to the soma for these channels to be active, whereas a weak depolarization could activate them further away. And so this helped propagate signals up the dendrites to the soma, which then acts as the counter. If you have enough depolarization, it fires an action potential, and that sends a signal to the next set of neurons. So this helped explain, again, some of the computational basics of what was going on in neurons as we started. And I think one of the things that really astounded Dan was that I managed to get all of this together and collect this data within the first 5 months of me being there. And so this publication was actually just 6 months after my arrival in the laboratory. And we ended up publishing 6 publications in total over the 2.5 years I was there. But that love of toys was really prevalent in Dan's lab. He was very positive, and he wanted me to succeed. And his biggest advice to me at this time was, if you want to get a job anywhere in North America, you should do another postdoctoral position at another big laboratory in the United States, because not many people are going to be aware of the University of Otago and what you did there. That's not a well-known university. So you need to go to a well-known place. So I did. And because I had this interest in computational neurobiology, I actually wrote to Terence Sanowski, who was at the Salk Institute for Biological Studies, and asked him if I could do a postdoctoral fellowship with him. Well, he was very enthusiastic about having me there, but it didn't take me long to find out that computational neurobiology wasn't for me. I didn't have the math skills that were necessary to succeed in this type of endeavor. And so that was a limitation of my education. But I still was able to adapt. And because of the work I've been doing in Dan's lab, I started filling cells so that we could get the cytoarchitecture of them, record that, and they could use this data to help them model. I could also fill the cells with or induce electrical activity in these cells and get parameters like how high their action potentials are, how wide they were, how often they would fire. This is, again, data that can be used for neurobiological modeling. And we also looked at their capacity for change. So these in particular are interneurons, which aren't supposed to show any form of synaptic plasticity. And we were able to show there are several types of interneurons that actually could show synaptic plasticity. And we ended up showing that one type of interneuron in the hippocampus actually wasn't an interneuron. It was a different type of pyramidal cell. And these are these giant cells. They have these two horns coming off them, so they're not a pyramidal cell. And they're actually located below the pyramidal layer in the CA1 region. So taking a different tact actually enabled me to contribute to computational neurology without engaging in it myself. And so this, again, was a very successful partnership. But even better, the Salk Institute was one of these places where a lot of exceptional researchers were already poised. And one of the individuals I'd met was this woman here, Henriette von Praag. And we had almost crossed paths a few years ago when we were both going to join a different laboratory together. But neither of us ended up going there. And one day she showed up in my laboratory at the Salk Institute and said, "Hey, Brian, how's it going? You know, we almost crossed paths and we didn't. And we've got this really interesting experiment going on in the laboratory." And she told me about this work by Gerd Kemperman, Georg Kuhn, and Fred Gage, where they had shown that you could make more new neurons in the brains of animals that are living in enriched environments. And she said, you know, it'd be really, cool if we could figure out why these animals make more new neurons, because they had bells and mirrors and toys and wheels and all kinds of things in these cages for the animals to play with. And the idea at the time was that it's this play behavior that was contributing to the production of new neurons. Well, so we went and looked at it, and it was very obvious that if you looked at what the animals spent most of their time, it was on running wheels. And so they spent a huge amount of time here and very little time playing with all the toys and all the other paraphernalia that our eyes were attracted to in the cage. And so we designed an experiment where we had animals with running wheels and animals without running wheels. We looked at their learning and memory capacity. And then we also did electrophysiology and immunohistochemistry on these animals. And what we found was that just exercise alone increased the number of new cells in the brains of these animals. So you can see here's a control animal. It's got a few red cells, which are the new cells in the brain. And then you have these animals that exercise, and they have many more red cells or the new cells in the brain. And what was really surprising to us was that these animals that exercise, so up here in this case, actually showed significantly more LTP than animals that didn't exercise. This increase in synaptic plasticity was specific to the dentate gyrus where these new cells were being produced, and you didn't see it in the cornu humanus, the area where you don't see new cells being produced in the brain. And so this was, some of the first work that gave the idea that new neurons actually contributed to the functional capacity of the brain. And so we published our paper, Running Enhances Neurogenesis, Learning and LTP in PNAS, another big journal, and shared first authorship on it. And my supervisor at the time, Terry Sinowski, and Fred Gage, who is Henriette's supervisor, were both the senior authors on the publication. One of the best things about this publication is it set me up for getting my own position. I ended up moving from here at a little short side journey, but ended up at the University of British Columbia, where I was brought in as a cohort of new investigators at the time. And this is Yu-Chin Wang, Wei-Hong Song, and my dear friend Alejandra, who unfortunately died soon after this picture. But I also brought my new family with me, and I had to find a time to balance my interactions with my science community at the university and my family. And this was another challenge that most people don't tell you about as you move through your career. But I found that time. I also ended up working more with Henriette and another individual, Alejandro Schindler. And we were supported by Rusty Gage, who was, again, another enthusiastic, supportive mentor who supported me even though I wasn't from his laboratory while I was starting my new laboratory. And we ended up using some of the techniques I learned from Dan's lab, only we made these new cells express GFP so that we could identify them. And then we went in and whole cell patch clamped them and looked at their electrophysiological properties and showed that they fired action potentials, that they received synaptic input, that they were capable of synaptic plasticity. And this proved that these new neurons actually became functionally integrated into the hippocampus and enabled learning and memory behaviors to occur. And other authors have taken up this work subsequently and show that they possess different types of channels that are more conveniently used for learning and memory processes. So I really, I learned a lot. I found my balance, and I started my own laboratory. And over the years, I think one of the biggest things I've learned is that you really gain a lot more by empowering those that are around you. And we have a lot of different avenues of research that have happened in my laboratory, from exercise to fetal alcohol spectrum disorders, Huntington's disease, Fragile syndrome, traumatic brain injury, patient-oriented research. And they're each carried by individuals who have their own passion for that type of work. You'll notice some of them are circled in bright yellow. Those are individuals from Brazil who came and contributed in my laboratory, and including Dr. Patricia Brocardo. This individual, JoAnne Gilmohapel, you may also know. She travels to USC quite often, from Portugal. Wishes she is from Brazil. Some of my students went on to become doctors. They're the ones that are circled in green. And some of them went on to other aspirations. And Luis is working in industry for a pharmaceutical company now, Luis Batillo. Heli Sickman, who actually worked with Patricia, is also working in industry. So they go into a lot of different careers, and they also drag me into another career. And that includes patient-oriented research. So Taylor and Jamie were just in Newski in February for the Floripa Neuro. And they're two of the main drivers that have helped bring my laboratory into looking at how humans can benefit from exercise and different cognitive training tools that we have, and how that can change biomarkers in their brain, which also promote synaptic plasticity. So empowering those around you can really be enriching, and really that's my end message, is that you can really live vicariously through the successes of your trainees. Just like I redesigned myself when I was no longer an athlete and became a scientist. Now I am moving from becoming a scientist to becoming a mentor, and I find that focusing on the successes of my trainees over the years has really brought me a lot of pleasure and kept me on my toes. They really stretch my bandwidth and make me learn a lot more. But it's really been an enjoyable experience, and I hope to continue it, for the next decade as we move forward. And there's a picture of the group from Lufsky then when I was there in 2023 for the first time. Right after I arrived, Patricia came up with a great idea to take us for a long hike. I'm sure some of you may remember that. But with that, I just want to say thank you for listening. The QR code actually links you to my laboratory webpage if you are interested in looking at any of the techniques or things that we do. My students just taught me how to do this, so I'm still learning, even at my old age. But anyways, thank you, and now I'm happy to take questions or to have you email me questions anytime at your convenience.
Hello, guys. Thank you so much, Dr. Christie. And then I say, like, I'm sure who is going to visit your website from your lab is going to feel really welcome. And then I could see it, but I was reading there. So we have actually two questions. I will read for you. The first one is from Bruna. I would like to thank Dr. Brian for his availability for the talk and for each lesson you taught us today. My name is Bruna, and I am PhD candidate, and I am being advised by Professor Deborah. In our study group, focus on neuroplasticity, and then I see that you have worked with different researchers. I would like to ask two questions. How important was the contact with other researchers for you, and how did we overcome to the challenge you mentioned in each lessons until you researched the point where you like to work?
It's in the early days, contact with other researchers wasn't that easy. You were very isolated. We didn't have Zoom, we didn't have computers, we didn't have email. In fact, I started talking with Dr. Johnston by writing letters that I mailed in the post office, and so the contact was much more difficult. But I think now with the advent of Zoom and meetings like this, that it's a much more useful way to engage with other researchers. And I often schedule Zoom calls in the morning, talking with other researchers who are doing different types of things, whether it's genetics or synaptic plasticity. I learn from them, they learn from me. And now it's, I think, much more important. And in the old days, you had to figure out on your own how to overcome problems, how to keep the slices warm. I made that little loop of copper around the objective to keep everything warm, right? There's a lot of innovation. But I remember a businessman once telling me, you don't have to reinvent the wheel. Chances are someone else has done something similar, and you can use the internet to find out how. And so I think using the internet now can really help you overcome a lot of those challenges. And most people are also very approachable. So I've seen an increase in individuals asking me, how do I cut my sections, or what stimulating electrodes do I use? But I also do that too. We ask other people what they do and how they do it. And so that interaction really helps you overcome any challenge. And, and to the point where, we even have my students go visit laboratories in Brazil, in Australia, in the United States, and we have students from everywhere come visit us as well. And that interchange, I think, is, one of the exciting things as a graduate student, a bit of travel, but it's really an opportunity to drive research into new areas that one individual alone wouldn't normally think of. The multitude will do more. Yeah.
Wow. Looking back to when you begin in your journal, we could see you worked without technology. I mean, without computer or cell phones. And now we have this question: in your experience, what do you think about the emerging technologies such as AI or advanced imaging techniques in advancing or understanding of brain function and plasticity in the near future? I think this is an interesting question because we're seeing the future right now, and then we can compare the past and the present, and how you imagine the future with the AI.
Yeah, and so the first image that I had of the serendipitous professor was actually one that was AI generated, and so that was me getting involved. And I'm lucky. I listen to my students, and One of my students is married to a computer scientist who, focuses on AI technologies. And, so I learned about DALL-E and ChatGPT, and now Gemini is what we use. So I use it a lot, but it makes me aware of its limitations. And so one of the things I was doing when I was making this talk was I was like, oh, I should really show people what, large irregular amplitude, small irregular amplitude, and theta activity look like. So I went to Gemini and said, generate an image showing LIA, SIA, and theta activity. And it generated 3 different waveforms. They were all exactly the same. None of them were SIA, LIA, or theta activity. So the accuracy wasn't there yet. So you do have to be careful. But if I was to say, what are some things I should talk about to a group of students to teach them synaptic plasticity, AI can give me a really good outline of things to cover to make sure that I'm hitting all the right points. We also use it extensively to generate our code, or to use with ImageJ to make sure that we're getting accurate results back and forth, right? AI can be a great tool to use, but it won't replace the thinking. You get in, you get out what you put in, right? So the more delineated your question is when you use AI, the better the information is you get out. But it's also limited. It only goes back so many years. It only can access things that are online and available. So there's pros and cons with it. But I'm always I don't think we should avoid things because they're new. I like new things, and I like using them as I can. And I think that also helps drive advances in imaging techniques, right? So I'm very positive, and I think AI has a great role for us ahead, and I think it will help us to devise even new crosses, for mice that have different fluorescent proteins that we can then image in different time points. So huge potential in future, and this is your future, which is so cool. Like, I'm excited for you guys, right?
Você quer fazer a pergunta? Eu posso fazer. Finalizando.
So I have just a question in some divagations, I think so. I don't know. It's in the past, neuroscience was not part of the researchers of nutrition researchers. And today we see a growing field, especially in studies on obesity. Even so, some students often do not see themselves in this context. It is as if studying neuroscience, I would distance them from nutrition. So in our research group, we are beginning the studies on the hippocampals and the context of obesity. And I don't know if you see something from these students, and do you think we are on the right track? Does it make sense to you?
Yeah, I mean, the most probably under-recognized, facet of neuroscience is the gut-brain axis, right? And it's starting to people are starting to pay attention to it now, but still not really understand it. And we've been working with a compound, adiponectin, which is produced by the burning of brown fat cells. And when adiponectins are released, it can bind to receptors in the brain, promote neurogenesis, promote synaptic plasticity. It can make you smarter. So that's when the end product of diet, right? The utilization of energy plays a role. But if you move back a step, how your gut uses energy is very much determined by your diet, and the bacteria in your gut, whether you're taking probiotics or not. This all influences the efficiency of this gut-brain axis, I think, and that's like a completely open field. We start, going back to some of the holistic medicine, and eating avocados is good, the fiber and the nutrition. There's so much potential there, but no understanding. So we can look at what nutritionists have done and what works for humans. And now we can actually sort of figure out the mechanisms by looking at the most efficient therapies and figure out how that changes maybe hypothalamic cells or production of new cells in the hippocampus, or where we have resistance to changing our eating habits because we have ingrained behaviors, right, that are reinforced, and how can we reset those? I have students that are also interested in, how you can reset the gut-brain axis by using psychedelics, and it's almost like with PTSD, you could also reset behaviors. You can, change appreciation of the oneness of the world. There's so many exciting things that are growing out of that field. So yeah, great question.
Thank you.
I think we have another question. Thank you, Dr. Christie. It was a great session to listening. Clarity and simplicity are truly remarkable and do not something on our encounters very often.
Encounters,
It's kind of common. And then I think we have time for the last question, which is for those who was just beginning their careers in neuroscience, particularly those interested in brain plasticity, what advice would you provide in terms for both academic growth and navigating and evolving
research landscape?
Yeah. So I mean, hopefully I came across, the follow your passion. You should, whether it's a relationship or your career, you should never, subjugate yourself to someone else's, drives and motivations. You should be following your passion, right? And there are lots of people out there who will help promote your passions. They understand, like, it's much better if I have a happy student, I have a hardworking student too, because they love what they're doing, right? And the productivity is much more. There's other professors that are out there that, and sometimes they're very famous professors too, who the show is all about them. And so the work is, all about the X lab or the Y lab. And they want the trainees only to focus on the projects that they want done. And that can work for some people. Like, it's not a bad thing. And it can be a pathway to success. But if you're not happy in that situation, if that isn't a happiness pathway for you, my advice is follow your passions, make the moves. I made the moves when I started. I changed my lab and my PhD. I advise graduate students in our program. I'm going back to be the graduate advisor again, and I advise them continuously. If you're not happy, it's okay to change labs. Like, it's okay. It's better for everyone, because your old supervisor will get someone who wants to do what they want to do, and you will get a supervisor who enjoys you doing what you want to do, right? So that's probably the biggest advice is make sure you're following your passion, and don't be afraid of reaching out. The worst thing that's going to happen if you reach out to a professor is they don't respond.
Yeah.
Right? They're not going to what is me, right? Worst thing is if they don't respond, and more than often than not, they will respond. I even try and find individuals who speak Farsi or Portuguese or French or German or whoever the student's from, so that we can talk. And I have a very international laboratory, and so oftentimes it's easier than you think to even communicate, even if you're not confident in English. It's okay.
I'm gonna give a comment here. For those who doesn't know it, Professor Brian, he was evaluating me in my poster. He's the first scientist and neuroscientist who was evaluating me, because it was my first post of my life.
Presentation
of my life.
And you kicked it.
I was like, oh my God, I'm talking to Professor Brian. That is my dream. So it was really a huge moment for me. And this is the second time that I'm meeting him. That I have a pleasure to learn from your expertise. And then I see that you're inspiring your lab, your students, and it's crossing the world, right? You arrived in Brazil, and then many other countries, and then we are really really happy just to stay with you today. And I don't know if anybody else has a question.
I think so. I think we have just not too much time.
Yeah, unfortunately.
Unfortunately, because it's a very, very good talk.
Yeah, it is.
And I like to say thank you, Brian. And that's something that I always say to my students. And for me, that's, I don't know if it's the same in Portuguese, is like the know you already have. So take your chance and try to talk to the researchers. This is a very important thing that you learn, including in your career. So thank you very much, Dr. Christie and Danielle. We will now take a break of 1 hour to have lunch. And after that, we will move to our second speaker for today. So to our audience, see you soon at 2 p.m. Thank you. Thank you very much.
Thank you so much. I enjoyed being here. Cheers.